Hi, I'm Dr. Sarah Beth Redmond. I'm a professor of biology, and I'm going to be talking about vaccines and herd immunity. I want to say up front that this is not medical advice. This is for educational purposes. I will be listing some resources in the description for this video. So if this is interesting and you would like to find more information, I encourage you to use those. I also encourage you to consult your medical clinicians as far as things that regard your own health and your health plans. So some starting vocabulary, an antigen is some sort of foreign substance that can induce an immune response, especially the production of antibodies. There is a subset that we would consider immunogens. So these are antigens that are going to actually induce a strong immune response instead of just tolerance. So as an example, we commonly refer to the blood group antigens, so A, B, O are antigens, but you don't make the antibodies to your own blood type, so it's not immunogenic for you. It might be immunogenic for someone else. A vaccine is a specific preparation of immunogens, and they are targeted to certain types of organisms or certain toxins in order to be very specified. So very different than what you would encounter if you were actually encountering those molecules in the environment. This is an isolated subset. And finally, herd immunity is the proportion of the population which needs to have an immune response in order to manage transmission rates. So when you look at antigens, they have variation in the size, complexity of what we call epitopes. And these are the areas where antibodies are able to recognize unique features of the antigen. So these parts in this example are pretty similar. They're not unique. They wouldn't identify that antigen, but there are places on that antigen which are more unique. And so those are the epitopes. We want to have epitopes that are very different from what the molecules in your normal body would be. We want to have epitopes that are fairly large. So some epitopes can actually interact with multiple different antibody structures. And we want to have a large number of epitopes so that there's an increased chance that one or more of your antibodies that your body is able to produce would actually bind to them. So when we look at the types of molecules that are able to be immunogenic, again, we want things that are big, we want things that are different from us, and we want things that are fairly complex. So things like amino acids are very small, they're not immunogenic. Lipids are fairly large, but they're not very complex, so they have low immunogenicity. Carbohydrates, here we're starting to get something that can be fairly immunogenic as long as it's different from what is in our body. Our body also has a lot of carbohydrates, so some of them won't work. Really where we're looking if we're trying to develop good immunity is proteins or whole organisms. So when we look at the ability to isolate specific proteins or specific organisms, that's where we would look for a vaccine target. Another factor with vaccines is the route by which they're administered to a host. So I've listed here some common ways in which this can be done. Many of the vaccines that you'll see that are distributed to large populations are intramuscular, subcutaneous, nasal distribution. And the difference here is how does your body actually encounter it? So something that's going to be inhaled through your nose is going to be encountered by a different part of your immune response than something that is intramuscular. And this affects the rate at which your immune response actually encounters it. So if we went directly into your circulatory system with an IV, then that gets encountered by the immune response much more rapidly than something that goes intramuscularly, where it has to be distributed throughout the muscle. Subcutaneous is even slower because it's going to have to be distributed into the musculature around the area. So these actually affect the rate at which the vaccine is encountered, in addition to stimulating different parts of the immune response. Finally, adjuvants These are molecules that can be co-administered with the antigen in order to increase the immune response. So you'll see things like salt solutions. These historically have sometimes been very nasty. There's one called Freund's Complete Adjuvant, which is not used in vaccines that are administered medically, but it is used in research. And it contains heavy metals, which can be very inflammatory. More commonly in vaccines that are distributed for medical purposes, you'll see things like larger proteins in order to have something that can attract lots of antibodies that could potentially also interact with the antigen. We also have things like receptor targets. So some of these are actually the molecules that your immune system would normally produce in order to induce inflammation. So if we include those with the vaccine, then the inflammatory process starts earlier. These can also be receptor targets for receptors on the cell surface to help the vaccine actually be taken into a cell. We can also have surface coatings. So some types of antigens are not very stable if we just put them into your body, or they might need a surface coating to help them get into the cell. So those are also fairly common. So what actually happens when you get a vaccine? The main thing that's going to be impacting you is inflammation. So inflammation will begin at the site where the vaccine was delivered. When you have an intramuscular vaccine, some of that actually comes from the damage to the tissue from the syringe and from adding extra fluid. But there's also going to be reaction to the adjuvants, general signals sent by your body to trigger inflammation. So what you'll experience is that at the site of the vaccine, inflammation can cause pain, can cause redness, can cause loss of function. So those are things that you might experience. As it gets distributed throughout your body, you are going to have the possibility of more systematic effects, things like headaches, fatigue, some people get nausea. These are all responses to the inflammatory process. Once white blood cells are brought in through that inflammatory process, they will take in the antigen, and they will interact with each other and the antigen to identify which versions of receptors they have that can actually bind to the antigen. And once those are identified, the number of white blood cells and antibodies will increase for a short period of time. So this graph I have over here is looking at antibody levels for two different types of antibodies, IgG and IgM. IgM is something that's produced fairly quickly, but it doesn't have the ability to be produced at a high rate. So you can see the IgG on a second exposure is going to peak at a much higher level. This is why you'll see some vaccines have boosters, because after the first response, which takes about 2 weeks, a second exposure is going to increase your ability to respond in the future. Okay, so how has the COVID-19 pandemic actually impacted our ability to produce vaccines? One of the big things has been the level of cooperation. And this is something that you don't necessarily see in a lot of particularly medical research. Industry research is very focused on finding targets that can be very productive for them financially. So it's not often incentivized to have cooperation. The other project that has been on a similar scale to this one was the Human Genome Project, which was completed in 2000. And if you look at that project, a lot of the framework that was established in order to promote cooperation during that project was actually really important for maintaining this framework of cooperation now. Another thing that has been really crucial was a transition to using mRNA vaccines. So it can be very costly to produce protein antigens or to produce organisms that are not infectious for a vaccine. So when we look at trying to develop a vaccine that is both specific enough and immunogenic enough, we have to balance those things. So we've got the spike protein, which I'll talk about in a minute, that's on the surface of the SARS-CoV-2, but it's fairly costly to make proteins. So we actually took a sidestep and said, well, you can take mRNA and make protein. So if we can make mRNA at a much more cost-effective route, then we can make the mRNA, put that into the cells make the protein themselves. This also has the immunological advantage that the protein is produced inside of the cell as opposed to outside of the cell. So instead of having it look like there's just virus kind of floating around in your body, it actually looks like your cells are infected. We also had a widespread testing and distribution plan that was discussed and developed by both international organizations and within different countries. And this really helped to speed the way in which the vaccine was able to be rolled out to populations. So what's actually in the vaccine? So this is broadly not referring to any particular brand. The mRNA codes for a surface protein called spike. This is the antigen. So the mRNA is not immunogenic itself, but once it goes into the cell and is used to make the spike protein, that protein acts as the antigen. The mRNA is then degraded, and the spike protein is also degraded. It doesn't have a long-term lifespan, and it is importantly not able to cause infection on its own. So this is not going to give you coronavirus. There are also lipids that are used to coat the surface of the antigen so it can easily enter the cells. These are the adjuvants. So if you think about cell membranes, there is this barrier for things to get into the cells. But if we include a similar lipid bilayer around the antigen, then that can serve as a mediator to allow the antigen to actually get in. So if you look at the ingredients list for the vaccine, these are the really complex looking names, and all these refer to the different positions of the chains and other connections between molecules in these lipids. These are all normal lipids. They are structurally the same type of molecules that you have in your cell membranes. They're also going to degrade over time. So again, this is not a long-term impact from the vaccine. And finally, there are some salts and one sugar to help stabilize the vaccine during transport. When you chill down molecules, that can impact their structure. So these help to maintain things like pH and salinity. These are all also molecules that would be normally found in your cells. So how is herd immunity determined? This is going to talk a little bit about the R0, which is the rate of transmission related to the duration of infection. Basically, this means once someone is infected, how many more people are they likely to infect? As some common examples, measles is one of the highest R0s that we observe. So what this means is that once one person is infected with measles, they're likely to infect 14 to 15 additional people. As you can see, SARS-CoV-2 has an R0 of about 2 or 3, depending on strains. There are some strains that have higher transmission rates that we've observed, so they would have a higher R0. The other thing is that not everyone has the same likelihood of transmitting the disease, and we call that value K. So K is impacted by things like population density. Do you live in a city or a rural area? The incubation period, so how long do you have the ability to be infectious before you recognize it? Variation in the response that people have to the infection. So some people don't ever actually show symptoms, so they don't know they're infected, they're carriers. Some people are super spreaders where they can actually be more transmissive than average, and also variation in behavior. So the K for SARS-CoV-2 has been estimated at between 0.04 and 0.2. What that means is that between 4% and 20% of the people who actually get infected are responsible for the transmission to other individuals. So most people are not actively transmitting. So these things interact, but the problem is they're not constant. The R0 can change as different strains emerge and also as we get an increase in vaccination, so who it can be transmitted to. But importantly, K will change as population behavior changes. So we've all been working with the idea of social distancing and masking in order to reduce K and reduce the number of people who are actually transmissive. But what we are going to see is that as people become vaccinated, they're going to want to reduce these precautions. Don't reduce your precautions, because that will actually go back to increasing the overall transmission. See the